I'd like to welcome to the stage our first contestant, Neha Bokil from the Department of Biology, giving her talk, Why Our Sex Chromosomes Matter.
How are male and female people different? The first thing that often comes to mind is clearly males and females have different body parts, specifically those associated with reproduction. But what if I told you that it's not just the reproductive stuff that's different, it's actually every single tissue in our bodies? This seems strange. We all have hearts, livers, immune systems. How could those things be different between males and females? But consider this. For every one male that has the autoimmune disease lupus, there are 9 females with that same illness. And that's just one example. We know a ton of diseases like Alzheimer's, COVID, heart disease, that show a significant sex bias in their incidence, symptoms, or mortality rates. There must be baseline differences between healthy males and females that predispose us differently to illness. Our lab asks the question, what are these baseline differences and where do they come from? Now, genetically, there is a difference between males and females, the sex chromosomes, X and Y. Females typically have 2 X's, while males typically have 1 X and 1 Y. But know that some folks are not male nor female, and as we'll see, some folks are neither XX nor XY. Regardless, all of our chromosomes are comprised of DNA, which encodes our genes, wrapped around structural proteins. These proteins are important because they can be modified in different ways to make some regions of DNA heavily condensed and silent, as I've shown with gray beads, or to make active regions open and accessible for the cell to read, like the blue beads. Both the X and Y encode factors that can regulate where the blue and gray beads are across all our chromosomes, but we don't know which genes are impacted by this. In my thesis work, I am identifying those genes. How do I do this? Remember, not everyone is XX or XY. It's possible for some people to have up to 5 total sex chromosomes. What I do is quantify the blue and gray beads across all genes in cells from these individuals with different numbers of sex chromosomes. This gives me the power to observe how each additional X or Y changes how silent or active a particular gene is. I've found hundreds of genes, like the one I've depicted here, whose blue and gray beads are affected by increased numbers of X's or Y's. And the impact of having more X's isn't always the same as the impact of more Y's. This has huge potential to cause genes to be read differently between XX and XY people, and thus cause differences between females and males. Hopefully, knowing which genes are affected by X and Y will help us better understand sex differences in disease and how to treat those illnesses. Thank you.